Evo2, the largest open-source AI model for biology. Introduction.
On February 19, 2025, the scientific community witnessed a groundbreaking advancement in synthetic biology with the announcement of Evo2, developed by a collaborative team from the Arc Institute, NVIDIA, Stanford University, UC Berkeley, and UC San Francisco.
Evo2 is heralded as the largest open-source AI model for biology, boasting an unprecedented ability to predict pathogenic mutations in genes and design entire eukaryotic genomes.
This comprehensive report explores the technical innovations, capabilities, ethical considerations, and potential applications of Evo2, along with its impact on the future of biological research.
Overview of Evo2.
Evo2 is a generative AI model trained on a staggering 9.3 trillion nucleotides derived from over 128,000 genomes across all three domains of life: bacteria, archaea, and eukaryotes, including plants and humans. This extensive training allows Evo2 to analyze genetic sequences on a scale previously unmatched,
facilitating rapid advancements in understanding and manipulating biological data. Key features and capabilities. 1. Pathogenic mutation prediction. Evo2 can accurately predict disease-causing mutations in human genes, achieving over 90% accuracy in tests involving the BRCA1 gene, which is significantly linked to breast cancer. 2. Genome design. The model can generate complete genetic sequences, including those of mitochondria and simple bacterial genomes. Its capability extends to designing DNA sequences specifically tailored for certain cell types, enhancing the precision of gene therapies. 3. Contextual understanding. Building upon its predecessor Evo1, which focused solely on single-cell genomes, Evo2 can process sequences of up to 1 million nucleotides. This allows the model to capture long-range interactions within the genome that influence gene behavior. 4. The technical innovations. Evo2 employs a novel architecture called StripedHyena 2, designed to efficiently manage longer sequences. This innovation allows for a larger context window, crucial for comprehending complex genomic interactions. Development timeline.
Evo1 launch. Released in the fall of 2024, Evo1 focused on bacterial genomes and demonstrated initial success in genetic analysis. Transition to Evo2. Between late 2024 and early 2025, researchers expanded Evo1's capabilities, training the new model on a vastly larger dataset. This rapid development culminated in the official launch of Evo2 on February 19, 2025. Technical architecture. Evo2's architecture, StripedHyena 2, represents a significant improvement over previous models. Traditional AI architectures struggled with the computational demands of processing long genetic sequences, as the complexity of relationships between nucleotides increased exponentially with sequence length. StripedHyena 2 mitigates this issue by using a combination of convolutional and attention mechanisms to efficiently model both short and long-range dependencies in genetic data.
Ethical considerations. In recognition of the potential misuse of such powerful technology, Evo2's developers implemented strict ethical guidelines. The training dataset intentionally excluded pathogenic viruses that infect humans and other complex organisms. Furthermore, the model is designed to avoid generating meaningful responses to queries related to harmful pathogens, ensuring that it is used responsibly within the scientific community. Use cases. 1. Disease research. Evo2's ability to rapidly determine whether genetic mutations are benign or pathogenic can significantly accelerate the pace of disease research, allowing scientists to focus on more complex questions. 2. Gene therapy development. Evo2's capacity to design genetic elements with cell-specific activation capabilities promises to advance targeted therapies, thereby minimizing unwanted side effects. 3. Synthetic biology. The model's genome design capabilities can aid in the engineering of novel biological systems, potentially leading to breakthroughs in bioengineering and environmental science. Potential implications for medicine and bioengineering.
EVO2's unprecedented scale and capabilities are poised to revolutionize the field of medicine by enabling researchers to make rapid progress in understanding genetic diseases.
The model's ability to predict pathogenic mutations and design tailored genetic therapies could lead to more effective treatments with fewer side effects. Additionally, EVO2 could facilitate the engineering of novel biological tools and systems, opening new avenues for research and innovation. Collaboration and development.
The successful development of EVO2 required extensive collaboration across multiple institutions.
The project brought together expertise from machine learning, biological sciences, and experimental biology,
creating a multidisciplinary team capable of tackling the complex challenges posed by such an ambitious endeavor. This collaborative approach not only accelerated the development timeline but also ensured the model's practical utility in real-world applications.
Conclusion. EVO2 represents a significant milestone in the convergence of AI and biology, providing researchers with a powerful tool to advance their understanding of genetic information and its implications for health and disease. By making this model fully open source, the developers have democratized access to cutting-edge technology, empowering scientists worldwide to leverage EVO2 in their research pursuits. As the field of generative biology continues to evolve, EVO2 stands at the forefront, paving the way for future innovations that could transform both medicine and environmental science. The content surrounding EVO2, the largest open-source AI model for biology, presents several counterintuitive pieces of information that challenge conventional understandings in both biology and artificial intelligence. Here are the key counterintuitive insights. 1. Predictive power versus experimental validation. Traditionally, biological research heavily relies on experimental validation to confirm hypotheses regarding gene functions and mutations. EVO2, however, can predict pathogenic mutations with over 90% accuracy based solely on computational analysis. This suggests that computational models can sometimes provide insights that are faster and potentially more reliable than lengthy experimental methods, which could revolutionize how scientists approach genetic research. 2. Open source versus misuse potential. The decision to make EVO2 fully open source may seem risky due to the fear of misuse for harmful purposes such as designing bioweapons. However, the developers have implemented safeguards to mitigate this risk, including excluding pathogenic viruses from the training data set. This highlights a paradox where the open-access nature of advanced technology can enhance collaborative research while simultaneously raising ethical concerns about potential misuse. 3. Longer context windows. EVO2's ability to process sequences of up to 1 million nucleotides is a significant technical advancement, allowing it to capture long-range interactions within the genome. This contrasts with older models that focused on shorter sequences and missed critical biological interactions. The notion that a larger context can lead to more accurate predictions runs counter to the traditional view that more data is always better, as the computation needed to analyze longer sequences can be exponentially greater. For a generative capability, EVO2's ability to generate entire genomes or specific gene sequences challenges the perception that biological engineering must strictly follow evolutionary pathways. It allows for the deliberate design of genetic sequences that may not exist in nature, suggesting that evolution is not the only pathway for genetic development. This capability raises philosophical questions about the nature of creation and manipulation in biology. 5. AI learning versus data memorization. The claim that EVO2 is not merely regurgitating learned data but rather developing internal representations of biological concepts is counter to the common critique of AI models as stochastic parrots. The use of a sparse autoencoder to interpret the model's internal workings suggests that EVO2 has genuinely learned from its training data, offering a glimpse into the underlying biological principles rather than mere memorization of sequences. 6. Speeding up evolution. The idea that EVO2 can speed up evolution implies that biological processes which typically take millions of years can be accelerated through AI. This challenges the notion that evolution is a slow gradual process and suggests that computational tools can facilitate rapid advancements in bioengineering.
7. Collaborative nature of development. The collaborative effort between multiple prestigious institutions Arc Institute, NVIDIA, Stanford, UC Berkeley, and UC San Francisco illustrates a shift in how complex scientific advancements are achieved. The expectation that groundbreaking developments must arise from isolated research teams is countered by the success of this interdisciplinary approach, emphasizing the importance of collaboration in addressing complex scientific challenges. 8. Generalist versus specialist models. Evo2's generalist design, capable of addressing a wide range of biological tasks, stands in contrast to the emerging trend of highly specialized AI models. While many fields advocate for models tailored to specific tasks, Evo2 demonstrates that a broad approach can yield significant insights across various domains of biology, challenging the idea that specificity is always preferable in AI applications. These counterintuitive insights underscore the transformative potential of Evo2 in the realms of synthetic biology and artificial intelligence, reshaping our understanding of both fields and prompting a re-evaluation of traditional methodologies and ethical considerations.
Significant content overlooked in the Evo2 report.
The Evo2 model represents a monumental leap in the ability to understand and manipulate biological systems through artificial intelligence. While much attention is rightfully focused on its capabilities, such as predicting pathogenic mutations and designing entire genomes, there are several layers of significant content that many might overlook. These aspects are crucial not just for the immediate implications of Evo2, but for the broader context of its development, usage, and future potential in synthetic biology. 1. Open source nature and collaborative development. One of the most significant yet potentially overlooked aspects of Evo2 is its fully open source nature. This facet is crucial for fostering innovation and ensuring that the model is accessible to a wide range of researchers, including those in underfunded or less resourced institutions. The collaborative effort among prestigious institutions like Stanford, UC Berkeley, UC San Francisco, and NVIDIA underscores a model of interdisciplinary research that encourages the sharing of knowledge and resources. Why it matters. Open source software has historically accelerated technological advancements by allowing users to build upon existing frameworks. This model can democratize access to cutting-edge technology, enabling researchers across the globe to contribute to and benefit from Evo2. It also mitigates the risks of monopolization of biotechnological advancements by private companies, which could otherwise hinder public health and scientific progress. 2. Technical innovations. StripedHyena 2 architecture. The introduction of the StripedHyena 2 architecture is another critical innovation that may not receive the attention it warrants. This new architecture allows Evo2 to process up to 1 million nucleotides at once, significantly enhancing its ability to understand long-range interactions within the genome. It combines various computational techniques to manage the challenges posed by longer sequences, a limitation that previous models struggled with. Why it matters. Understanding the genome is not just about the sequence of nucleotides, it's about how those sequences interact spatially and temporally within a cell. The ability to analyze long-range relationships can lead to breakthroughs in understanding complex genetic diseases where multiple genes and regulatory elements are involved. This aspect of Evo2 could pave the way for more accurate models of gene regulation and expression, thus informing better therapeutic strategies. 3. Ethical and safety measures. The ethical considerations surrounding the use of Evo2 are of paramount importance. The developers have implemented several safeguards, such as excluding pathogenic viruses from the training dataset and ensuring that the model does not respond to queries about harmful pathogens. These measures are designed to prevent misuse, such as the creation of bioweapons. Why it matters. In an era when synthetic biology is rapidly advancing, the potential for misuse grows proportionally. By prioritizing biosecurity, the developers of Evo2 are setting a precedent for responsible AI use in biotechnology. This proactive stance is essential for maintaining public trust in scientific research and ensuring that advancements in this field benefit society rather than pose risks. 4. Speed of research and discovery. EVO2's ability to predict pathogenic mutations in mere seconds compared to the months that traditional methodologies typically require may be underappreciated. This rapid analysis not only accelerates the pace of genetic research but could also significantly impact clinical diagnostics and personalized medicine. Why it matters: the speed of research is crucial in the context of emerging diseases and genetic disorders. For example, during the COVID-19 pandemic, rapid genetic analysis was essential for understanding the virus and developing vaccines. EVO2 could similarly expedite the discovery of genetic links to diseases, allowing for quicker responses in clinical settings and potentially saving lives. 5. Implications for environmental science. While much of the focus on EVO2 is on its applications in medicine and genetic research, its implications for environmental science and bioengineering are equally significant. The model could be used to design microorganisms that can, for instance, degrade pollutants or produce biofuels, addressing some of the pressing environmental challenges of our time. Why it matters: the intersection of synthetic biology and environmental science holds immense potential for sustainable solutions to global challenges. By harnessing EVO2's capabilities, researchers could develop innovative strategies for environmental remediation, conservation, and sustainable resource management, ultimately contributing to more resilient ecosystems. 6. Long-term vision and future applications. The researchers envision EVO2 as a foundational tool that could lead to the development of more specialized AI models tailored for specific tasks within biology. This vision hints at a future where AI-driven tools can tackle a plethora of biological questions, from understanding complex traits in agriculture to designing new organisms for specific industrial applications. Why it matters: as EVO2 serves as a springboard for future innovations, it emphasizes the importance of foundational models in AI and biology. The adaptability of such models could revolutionize how we approach biological research, shifting from reactive trial-and-error methodologies to more proactive predictive strategies. Conclusion: EVO2 is not just a technological marvel, it embodies a paradigm shift in how we understand, manipulate, and innovate within biological systems. The aspects discussed: open source collaboration, technical innovations, ethical considerations, rapid research capabilities, environmental implications, and long-term vision, are critical to grasping the full significance of this development. By appreciating these elements, stakeholders can better navigate the ethical landscape of synthetic biology and harness the capabilities of EVO2 to address some of humanity's most pressing challenges in health and environmental sustainability. 1. Pathogenic mutation prediction. EVO2's ability to predict which mutations in human genes are likely to be pathogenic is a groundbreaking feature. In tests involving the BRCA1 gene linked to breast cancer, EVO2 achieved over 90% accuracy in identifying disease-causing mutations. This capability significantly accelerates the traditional process of determining the pathogenicity of genetic changes, which usually takes months of laboratory experimentation. By providing rapid and accurate predictions, EVO2 could streamline genetic research and clinical diagnostics, leading to faster identification of genetic disorders and more effective treatments. 2. Genome design and bioengineering applications. EVO2 can design entire genetic sequences, including those of eukaryotic organisms, which marks a significant advancement over existing models. By generating DNA sequences tailored for specific applications such as gene therapies that activate only in certain cell types, EVO2 enhances the precision of genetic engineering. This capability opens new avenues for targeted therapies, reducing potential side effects and improving treatment effectiveness. The model's ability to handle sequences of up to 1 million nucleotides enables it to consider long-range interactions within the genome, a critical factor in understanding complex biological processes. 3.
Innovative AI Architecture StripedHyena 2. The development of the StripedHyena 2 architecture is a key technical innovation that enables EVO2 to process longer genetic sequences efficiently. This new architecture allows the model to maintain a broader context window and analyze relationships between distant genomic elements. The increased computational efficiency reduces the challenges typically associated with long sequence processing, allowing EVO2 to derive insights from the complex interdependencies present in eukaryotic genomes. This advancement not only improves predictive accuracy but also enhances the model's applicability across a wider range of biological research. 4. Ethical considerations and open-source accessibility. EVO2 is fully open source, which means its code, training data, and methodologies are accessible to researchers worldwide. This transparency fosters collaboration and innovation within the scientific community. However, the developers have also implemented ethical safeguards to prevent misuse, such as excluding pathogenic viruses from the training data and ensuring that the model does not generate harmful sequences. By addressing these ethical concerns while promoting open access, EVO2 represents a balanced approach to advancing scientific research in a responsible manner. This commitment to ethical development is crucial in the context of powerful technologies that could potentially be misused in bioengineering. 1. The Human Genome Project (HGP), launched in 1990 and completed in 2003. The Human Genome Project was an international scientific research initiative aimed at mapping and understanding all the genes of the human species. It involved sequencing the entire human genome, which consists of over 3 billion DNA base pairs. The HGP provided foundational knowledge for modern genetics, enabling discoveries related to genetic diseases, gene therapy, and personalized medicine. The project set the stage for using genomic data in clinical applications. Similar to how EVO2 aims to enhance understanding and manipulation of genetic information. 2. CRISPR-Cas9 gene editing. Developed in the early 2010s, CRISPR-Cas9 is a revolutionary gene editing technology that allows scientists to alter DNA sequences and modify gene function with unprecedented precision. This tool enables targeted changes to the genome, such as correcting mutations that cause diseases, creating genetically modified organisms, and advancing our understanding of gene function. The implications of CRISPR are akin to those of EVO2, which also aims to facilitate advancements in genetic engineering and therapeutic interventions. 3. AlphaFold by DeepMind. Released in 2020, AlphaFold is an AI program developed to predict protein structures based on amino acid sequences. It uses deep learning techniques to determine the 3D shapes of proteins, solving a long-standing challenge in biology known as the protein folding problem. AlphaFold's ability to accurately predict protein structures has significant implications for drug discovery, understanding diseases, and bioengineering, paralleling EVO2's promise to revolutionize genome design and mutation prediction in biological research. Future outlook for EVO2: transforming biology through AI. As we look ahead to the future of synthetic biology and AI, the launch of EVO2 marks a pivotal moment. This advanced open-source AI model, developed by the Arc Institute, NVIDIA, and leading academic institutions, is set to revolutionize our understanding and manipulation of genetic information. With its ability to predict pathogenic mutations and design complete eukaryotic genomes, EVO2 promises to open new avenues in medicine, environmental science, and biological research. Here are key predictions for the future impact of EVO2. 1. Accelerated medical breakthroughs. EVO2's ability to accurately predict pathogenic mutations, demonstrated by its over 90% accuracy in identifying harmful BRCA1 mutations, will significantly speed up the process of diagnosing genetic disorders. This will facilitate earlier interventions and personalized medicine tailored to individual genetic profiles. As EVO2 is integrated into clinical workflows, we can expect a decrease in the time required for genetic analysis, transforming how genetic counseling and testing are conducted. Furthermore, its genome design capabilities will enable rapid development of gene therapies. The ability to create DNA sequences that can activate or suppress genes selectively in specific cell types could lead to more effective treatments with fewer side effects. This precision medicine approach can be particularly beneficial in treating complex diseases like cancer, where understanding the intricate interactions between genes is crucial. 2. Innovations in drug development. The pharmaceutical industry is likely to undergo a transformation with the advent of EVO2. By enabling researchers to predict interactions between genes and potential drug targets, EVO2 will streamline the drug discovery process. The model's capacity to generate novel genetic sequences could lead to the development of new biomolecules such as proteins or enzymes that can serve as therapeutic agents or drug delivery systems. As EVO2 becomes a staple in drug development pipelines, we may see a significant reduction in the costs and time associated with bringing new drugs to market. The ability to simulate and test genetic modifications through AI will complement traditional laboratory approaches, allowing for more informed decision-making in drug design. 3. Environmental applications. EVO2's capabilities extend beyond human health into environmental science. The model can be utilized to design microbes that can efficiently break down pollutants or produce biofuels. By generating optimized metabolic pathways and genetic circuits, researchers can engineer organisms for bioremediation or sustainable energy production. As climate change and environmental degradation continue to pose significant challenges, EVO2 will play a role in developing innovative solutions. The ability to generate tailored organisms for specific environmental tasks will enhance our capacity to address ecological issues and promote sustainability. 4. Ethical and regulatory considerations. The open-source nature of EVO2 raises important ethical considerations. While the model enables groundbreaking research, it also poses risks of misuse. Developers have implemented measures to prevent the generation of harmful sequences, but the potential for biosecurity threats remains a concern. Moving forward, establishing robust regulatory frameworks will be essential to ensure responsible use of EVO2 and similar technologies. As the scientific community explores the boundaries of genetic engineering, discussions surrounding the ethical implications of creating new life forms or modifying existing ones will become increasingly prominent. Responsible stewardship of such powerful tools is paramount to avoid unintended consequences. 5. Interdisciplinary collaboration. The success of EVO2 underscores the importance of interdisciplinary collaboration in advancing scientific research. Future developments will likely see even more partnerships between computational biologists, bioengineers, clinicians, and ethicists. Such collaborations can foster innovation and ensure that diverse perspectives are considered in tackling complex biological challenges. As researchers leverage EVO2, we can anticipate a surge in collaborative projects aimed at exploring the vast potential of synthetic biology. This collaborative spirit will not only enhance scientific discovery but also drive the translation of research findings into practical applications. 6. Personalized genomics and healthcare. With the capabilities of EVO2, the future of genomics will lean heavily towards personalized solutions. Individuals may soon have access to tailored genetic assessments that provide insights into their unique genetic risks and predispositions. EVO2's predictive analytics can guide decisions regarding preventative measures and lifestyle adjustments, ushering in a new era of personalized healthcare. Moreover, as genomic data becomes more integrated into healthcare systems, we may witness the emergence of AI-driven platforms that offer real-time analysis of genetic information, empowering patients and healthcare providers to make informed choices. Conclusion: EVO2 is set to revolutionize the fields of synthetic biology and genomics, ushering in an era of unprecedented discoveries and applications. Its ability to predict mutations and design genomes will accelerate medical breakthroughs, enhance drug development, and enable innovative environmental solutions. However, The responsible use of such powerful technology will be crucial to ensuring that its benefits are realized without compromising safety and ethical standards. As we navigate this exciting frontier, the collaborative efforts of scientists, ethicists, and policymakers will shape the future of biology in profound ways.